I now have the great pleasure of introducing Dr. George Brainard. Dr. Brainard joined the Jefferson faculty in 1984. During his career, Dr. Brainard has taught neuroscience to over 10,000 medical and graduate students. He has received the Lindback Foundation Award for his distinguished teaching. His painted portrait was dedicated to Thomas Jefferson University by the graduating class of 1989. He is professor of neurology and director of the Light Research Program at Thomas Jefferson University. His academic work has been concerned with the effects of light on biological and behavioral responses of animals and humans for over 40 years. For the last 10 years, he and his colleagues at Harvard Medical College have assisted NASA in the development, testing, and implementation of the dynamic lighting system that has been installed on the International Space Station. His research has been supported by grants from the FDA, the National Institutes of Health, the Department of Energy, and many other sources. Dr. Brainard has authored more than 100 scientific articles and contributed to more than 50 books. He is an honorary lifetime member of the Yoga Research Society. His topic is exploring light in inner and outer space. Please join me in welcoming Dr. George Brainard.
Well, thank you for that introduction, Linda, and I also really want to thank Dr. Pratap for inviting me to speak with you this afternoon, Barbara Levitt, who has handled so many of the details, and all of the Yoga Research Society staff that has worked hard on the next 2 days event. My goal in this time period is to touch on a number of things: inner light, the third eye, pineal physiology, space travel, candles, and some other stuff. I'm going to try to leave 15 minutes at the end for question and answers. I also want to acknowledge that I'm very pleased that my family was able to join us this afternoon. My wife Lane is here, my daughter Lindley and her wife Lily is here, and Dylan, my other daughter, is here. So thanks for coming in, and I'll show you some things you haven't seen before. As Linda mentioned, nothing really happens in research unless you have funding to make it go. And we're very grateful to have had a fair bit of federal support. The majority of our work has been supported by the federal government. But industry has also contributed in philanthropic support as well, philanthropic. My interest in yoga began as a teenager, and I'll comment on that era in a moment, but a young teenager. And in the community I lived in, there were no yoga classes. Pretty much everywhere in America right now, there's multiple yoga classes. There's only one Yoga Research Society, by the way, classical yoga as taught by Dr. Pratap. But we lived rural, and I'll show you that in a moment. And so I got mail-order yoga classes from the Self-Realization Fellowship, Paramahansa Yogananda, and that was my beginning. Then, when my family and I moved to Philadelphia, we met this guy who taught you this morning, and we've been great colleagues ever since. You see the cover page, front piece from the 40th Research Conference. I was honored to be invited to talk in that, and here, 10 years later, back again at it. So, you know, I know, this morning you were guided through a good number of postures and practices, and this is the core of classical yoga. And there's nobody that I know of that really instructs at this same level. Not that the other forms of modern yoga are bad or anything like that, but this is much to my preference. So, this notion of inner light. As you'll see, we study light in our laboratory, and that's physical light. But then there's that thing, metaphysical light, the light that's within us. And that interests me every bit as much as physical light. So where do we learn about that? Well, the great books of all major religions are very clear. Going back to the Vedas, from ignorance lead me to truth, from darkness lead me to light. This idea of light and truth is inherent there in the Vedas in many different ways. In the Old Testament of the Bible, and God said in Genesis, let there be light, and there was light. In the New Testament, John, light shines in the darkness, and the darkness has not overcome it. In the Quran, O mankind, there has come to you a conclusive proof of your Lord, and we have sent down you a clear light. And one of my favorites, and I'm not going to just go through all of the major world's religions, from the Tao Te Ching, can you cleanse your inner vision until you see nothing but the light? An open question. So I admire those sources of knowledge. I've felt they've informed me in my life, and I'm grateful that we have them. But I also am a scientist, and one of my favorite scientists is shown here, Albert Einstein. And his singular accomplishment, he accomplished many, many things during his lifetime, and he was a great humanitarian. But the general theory of relativity, and you see a page from his notes here at the bottom, which now you would find it in a library book and printed and so forth. A core element of that is what we're able to know about in the physical world, the composition of our known universe. 5% of it he called ordinary matter. That you can know it. You can touch it, like I touch this podium, you can see it, you can hear it, you can taste it. 5% of what's there in the universe is knowable to we human beings. Then there is 25% dark matter. Modern physicists have calculated this and confirmed that Einstein was right, and 70% is dark energy. We can't know anything about that other 95%. It's outside of our reach. We can theorize about it. We know it's there. Physicists have proven over and over again that stuff is there, but we don't know what it is. We don't know how it works. We really don't know a whole lot. So, you know, scientists are supposed to know things, and it's a little frustrating when the majority of everything in the universe is out of our reach. But it leads us to a very clear conclusion. And this is just a different representation of that. 5% ordinary matter swirling around out there in the cosmos, as well as on planet Earth, as well as within our bodies. 25% dark matter and 70% dark energy. It leads to the conclusion, we know that we don't know. Anything that we would presume, anything I would presume to say to you today with certainty, fundamentally, it's within that domain of we know we don't know, because we might discern little facts and little truths, but it's surrounded by things that are just completely unknowable. No despair. It's a safe place to live knowing that you don't know. This is a photograph from central Indiana, in a region that I spent part of my childhood in. And as you can see, cornfields, rolling hills, very, very rural. And that's where I spent some of my most formative years. The nearest town was Traders Point, Indiana. And from our home out in the cornfields, I could bicycle there. It was about 3 or 4 miles, so I could get to this town. This town had 1 small grocery store, 2 churches, and a grange hall. That was Traders Point. It was, but it was a destination because when you're out there in the fields, You know, sometimes you just want to see the big town. This is in the era of 1950s. Mostly what you saw were cornfields. However, my parents subscribed to Life magazine. I'm just wondering, am I dating myself? How many of you know of Life magazine? Oh, quite a lot. Okay. So that came, and to turn my attention to things other than the cornfields, I pretty much read Life magazine cover to cover. And this shows you one of the issues around the time that this magazine altered the course of my life forever. There was a very short paragraph, maybe one or two paragraphs, and a picture of the western fence lizard, the Sphenodon occidentalis. And so I read that little bit. Here it is. Scleroporus occidentalis, western fence lizard, didn't live in Indiana. It's much more out west. There were lizards in Indiana, of course, but there was this one that was featured in that teeny little article. And the thing that just totally captured my imagination was the western fence lizard, like we humans, has a lateral eye here, and you can see the hood of the other lateral eye, two eyes on either side of its head. But the fence lizard has an anatomical third eye, different than the two lateral eyes. And that notion that a living being on planet Earth had three eyes just was an upheaval to me. It captured my imagination. It still captures my imagination. So there is an individual, Richard Eakins, a zoologist who studied these creatures, western fence lizards, and other species that have third eyes. And this is a photograph taken from his book. Here you're looking down on the surface of the fence lizard's head. Here's one eye over here and one eye over there. And then he's drawn a circle right here on the dorsal surface of his head to illustrate that there's a small scale that's translucent. And that scale is positioned over a very rudimentary eye that has a lens and a retina, like our facial eyes have. And here's a diagram of it. Here's the skull. Here's that translucent scale over the what's called the parietal eye. It has a retina and a lens. It's not as fully developed as the lateral eyes, and it is right next to this structure, the pineal gland. So it was a twofer, third eye and pineal gland. Now, I was young, and I thought that was amazing. And then I thought to myself, I wonder what a gland is. Never heard of a gland. So went to the big dictionary, pulled it off the shelf, looked it up, and it said, a gland is a ductless organ. And I said, I wonder what a duct is. So I began this adventure of trying to learn a little bit about these structures, the third eye, the pineal gland. And it's interesting, you'd think this is an anomaly, right? Oh yeah, okay, so a lizard out west has a third eye. Fact is, the majority of vertebrate species have some form of third eye in their head, going from primitive fish like the lamprey eel to modern bony fish, over to amphibians, to lizards like the western fence lizard, even birds. So the majority of species with a vertebral column have third eyes. Then the branch that took off in the direction of mammals, where we live, you know, small mammals like mice, bigger mammals like primates, don't have anatomical third eyes like the majority of other species on the planet Earth that have a backbone. So Dr. Eakins here, this is from his book, has dressed up as Charles Darwin, and he's painted a circle on his forehead to show where, if we as a species had retained an anatomical third eye, where it would be located, similar to the other vertebrate animals. But of course, for him, this was fun, having good fun with his science. And then if we look at the human brain, and we think about the location of the pineal gland, Here is the human pineal gland right here, buried deep within the brain, not anywhere near the dorsal surface. It's not even a candidate to have a structure as a third eye. It's a gland, and a ductless one at that. And it's fairly close to where the pituitary gland would be over here. And that's where our species is. However, as we look at examples collected from wide range of sources, here is a reclining Buddha, and you see a raised urna on his forehead, which was purported to have occurred after his enlightenment under the Bodhi tree. So something akin to a third eye structure, perhaps. Here is the Tibetan calendar held by a bear god, and you see two dorsal eyes, or two lateral eyes, and a third eye perched on its forehead. This is a Sanskrit chakra tapestry, and brow chakra, crown chakra. Brow chakra is often coordinated to the third eye, but sometimes it's vice versa, as I'll show in the next slide. Here is an overlay of the chakras, crown, which in this version is associated with the pineal, and the brow chakra is associated with the pituitary, and the other chakras are coordinated with other ductless glands. So I find a disparity as I go through different sources of who says the third eye is the pituitary, who says the third eye is the pineal, but they're always one or the other, and again, it's source dependent, and perhaps it's changeable. This is one of my favorite pieces of artwork from Solomon Wolfing. You see this image of a lizard, and that's where I started my little journey with the third eye. You see the lizard, and it's superimposed on the head of a sleeping woman, and then above that you see the woman is awake, and there's no third eye, but there's illumination. So, metaphorical, physiological, we know we don't know. So whatever happened to Traders Point, Indiana, you're wondering? You weren't really wondering that, but I'm going to tell you anyway. Behind our house, there was a creek called Eagle Creek, and it was a very manageable creek. In dry seasons, you could walk across it on your feet. In rainy seasons, you'd get a rowboat and go across Eagle Creek. But that was Eagle Creek, and that ran behind my house. After we moved from Indiana to Cleveland, they dammed up Eagle Creek, and they made Eagle Creek Reservoir, which you see here. This is modern times. And Traders Point's underwater now. So Ben Warfield helped me make these slides, and he commented, he said, is this really where you grew up? And I said, yeah. And he said, there's something very haunting about towns that are now underwater. And it just sent a chill through my body. Hadn't really thought of it that way, but there you have it. So, I know you're saying, where's the science-y part of this? I came to hear about the science-y part, and so I'm going to go in that direction now. And our laboratory has a fundamental philosophy, which is, it's summed up as follow the photon. Photons are particles of light. And what we'd really like to understand is how the human eye absorbs photons. We all know it allows us to see the world, right? But we're interested in how did those photons trigger biological and behavioral change in human beings. And really, that summarizes the majority of the work that the government and industry and philanthropy supports us to do. So this next is the simplest rendition I can come up with. And I'll explain it, and we won't do any more than one more of these kinds of slides. Light comes into the human eye. It goes through the cornea, the lens, the vitreous humor, hits the back of the retina here. The retina are where the photoreceptors are. That's the first place photons are detected in the human eye. Signals are sent from the retina to a part of the brain, the thalamus, the lateral geniculate nucleus, and then relayed from there to the visual cortex at the back of your head, your occipital lobe. So that pathway is how we detect light to see the world, to navigate the world, to do our work, to enjoy interacting with one another. And we're really a strongly visual species, and this part of eye physiology has been studied for quite some time and is pretty well fleshed out. In more recent years, a new pathway was discovered, shown here, that projects not to the visual system, but into the hypothalamus. And you're saying, what do you mean the hypothalamus? It's labeled SCN. Well, that nucleus is the suprachiasmatic nucleus. It's easier to fit SCN into a little circle than suprachiasmatic nucleus. But the suprachiasmatic nucleus is an internal oscillator. It's, if you took it out of the brain and put it in a petri dish, and they've done this with rodents, but not with humans yet, but it will oscillate on a period of about 24 hours in the petri dish, disconnected from the brain. So it's an internal oscillator. It's the fundamental component of the biological clock, or the more technical term is the circadian system. And that's not a visual structure per se, but it in turn is connected to broad areas of the nervous system. And again, this is simplified. Projections go all the way down to the spinal cord, and I show you that in a moment, to the midbrain, the hypothalamus, the limbic system, other regions of the thalamus. And there's a bit of crosstalk between the visual system and the circadian system. So, ladies and gentlemen, SCN stands for suprachiasmatic nucleus, and that will be on the quiz. So I want you to remember that one. What is the suprachiasmatic nucleus doing? It's keeping track of biological time. It's receiving information about light and darkness in the surrounding environment, and it's in turn communicating that broadly through the nervous system so that the body can know what time it is and the status of light and darkness in the surrounding environment. So now I want to talk about melatonin, and here you're seeing the chemical structure of melatonin, and that's it for all species, including ours. And the simple notion is melatonin is a hormone secreted by the pineal gland, one of my favorite glands. Did I happen to mention that yet? It's secreted by the pineal gland. It gets out into the plasma, saliva, urine, cerebrospinal fluid, and so forth. And we secrete that hormone in a rhythm. The hatched bar shows you the period of darkness. So during the daytime, right now, if we had a phlebotomist, a nurse come and collect a blood sample or a saliva sample from you, and we measured it up in our lab across the street, it would be very low to non-detectable. You're not producing, your pineal gland doesn't produce melatonin in any substantive amount during the daytime. But with the onset of darkness, there is a rapid increase of synthesis and secretion that's maintained through the majority of the dark period and comes back down. The same pattern is observed in other species. So going down the so-called phylogenetic tree to a quail, high melatonin during darkness, low melatonin during daytime. A goldfish, high melatonin during darkness, low melatonin during the daytime. A moth, an insect species, high melatonin during darkness, low melatonin during the daytime. And this last one's a kicker. I wonder if anybody here knows what this is. Just shout that name out if you recognize that species. What was it? I couldn't hear. Yeah, this is a single-cell phytoplankton, Goniolix pulledra. It's ubiquitous through all of the oceans across the planet Earth. It's a single cell. It produces high melatonin at night and low melatonin during the day. Let's just think about that. It's a single cell. It doesn't have an eye. It doesn't have a pineal gland. It produces the same signature of high melatonin during the night, low melatonin during the day. So evolution has conserved this molecule, both in the structure of the molecule and its physiological expression within the species, whether that species is nocturnal, diurnal, or crepuscular. So it's a remarkable hormone all on its own, but let's focus on our species. Now, this image is collected from a very famous medical journal called the New York Times, and it's been modified slightly for detail and accuracy, but I'm going to walk you through this, and I promise, I think it's true. This is my last neural pathway of the talk. So, here you see light in the environment, enters the eye, stimulates the retina. A primary optic tract goes over to the thalamus, the lateral geniculate nucleus, and then to the visual cortex, allowing to see the world. You can see the slides, you can see me waving my arms around and so forth. Then there's a separate pathway, the retinohypothalamic tract, going to one of your favorite new nuclei, the suprachiasmatic nuclei, and that's that clock structure inside the nervous system. And then there's a short projection to a part of the hypothalamus called the paraventricular nucleus, and then it is a long axon that stretches all the way down to the spinal column, thoracic 1 and 2. Synapse is there, leaves the central nervous system to go synapse in the superior cervical ganglion, and then the postganglionic neurons climb the vasculature back up into the cranium and ultimately innervate the pineal gland. So that long, indirect, multisynaptic pathway is doing one thing. It's bringing information to the center of the brain about light and darkness outside of the body and biological time. And in turn, the gland secretes melatonin, and it has a crisp day-night cycle under ordinary circumstances, so high at night, low during the day. And then if you interrupt the period of darkness, if you shine a bright enough light into a person's eyes in the middle of the night when their pineal gland is producing lots of that hormone, there is a rapid suppression. So during exposure to bright light in the middle of the night, you'll cause a rapid suppression of melatonin. And we use that as a tool. And one of the ways we've used it as a tool is to ask the question, if bright white light in the middle of the night can suppress melatonin, what about the different wavelengths of light or the different spectrum of light? As we go through the visible spectrum from the short wavelengths, you're in the violet, indigo, blue, going to the middle wavelengths, greens and yellows and oranges, and finally the long wavelengths of red. So are they all equal, or is there a difference between them? Which ones act most like white light, or do you really need the full complement of white light to achieve suppression of melatonin? So we were fortunate to do a study on this, and this illustrates the equipment that we developed and used. We had a high power supply connected to an igniter. That igniter was in turn connected to a xenon arc lamp. Xenon arc lamps produce really intense white light, so intense that if you turn the light onto a piece of wood, it sets it on fire, right? And that's not the experiment, but we did that because it's fun. We then took that beam of intense white light and we channeled it into a device called a spectroradiometer. Then we selected down to very narrow wavelengths of light and bounced it into a big dome called the Ganzfeld dome. And that light beam then bounces around the dome, and if you have an individual sitting with their head held steady outside the dome, they'll see patternless light of whatever wavelength you've selected. And here it's being illustrated as a blue light. And we do this experiment in the middle of the night when their melatonin levels are high. And so for a normal, healthy man or woman to stay awake for a 90-minute period staring at patternless, homogeneous blue light or green light or red light, there's a tendency to fall asleep. So at the back end of the dome, we had a miniature camera attached to a monitor that allows us to see that their eyes are being held open because we're trying to understand the dose of light coming into the eye. We want that to be consistent and accurate. And so you see my colleague, Jeff Greason, who's in the audience today. There he is, he's waving at me. Jeff is tilted his head a little bit outside the dome so you can look in and see the green light that he's looking at, patternless green light. And then you can see Jeff's eye on the observation monitor. And you'll notice, you might be able to see it, you might not, his pupil is completely dilated because part of this experiment is before we do this light exposure, a person comes into the lab, they sign their consent forms, we dilate their pupils so their pupils are not going to interfere with the dose of light. And then they get a period of darkness, a period of light exposure, collecting blood samples during that time, and then ultimately they go home for the night. And that's the experimental step. I'm going to say something now. I'm going to break all the PowerPoint etiquette rules in the next slide. I'm going to show you the results of over 600 experiments done across 7 years, all on a single slide. That way we can kind of blow past them and it'll be a relief. They're all published and you can have them if you want them. Here they are. We studied 72 healthy men and women over a 7-year period. Groups of them were exposed to different wavelengths of light, as low as 420 nanometers, as high as 600 nanometers. So 420 is in the violet range, 440 is in the indigo range, 460 is blue, and so forth. So we collected this information looking at plasma melatonin percent change scores. So we're looking at that endpoint of the pineal gland, melatonin. That's why I say melatonin for us is a very valuable and useful tool. And so, but what do you do with all that? What you do with that is that's the foundation for a thing in the field of photobiology called an action spectrum. You take the half saturation constant from each one of those individual 9 graphs and you plot them on a single curve, and it's a much simpler rendition. Here it is. So each one of those black dots is derived from one of the curves on the prior slide. And then we curve fit to a thing called an Opson nomogram. And you can see it's got an R-square value of 0.91. That's a really strong coefficient of correlation. That gives you confidence that this curve accurately describes those data. And you'll see at the top the number 464 nanometers, and that is the mathematical peak of that curve. And that's important, and I'll say why in a moment. However, our colleague, Jeff and my colleagues, Mark Rolog, said, yeah, sure, well, that's a mathematical peak. But human biology is messy. There's a lot of variation. We can't just say a single number and say that with confidence. He said, so we added that if you do plus or minus 1 standard deviation, that peak is somewhere between 446 nanometers and 477. Again, these data, maybe they don't shout upheaval to you, but they did to us. We figured our careers were over. We were going to have to tell the world in the year 2000 that there was an undiscovered photoreceptor in the human eye, and people had been studying the human eye for so long, it was absurd that there would be a photoreceptor overlooked. But that's the conclusion that we had to come to. We published this in a strong journal, Journal of Neuroscience, and this is the title of the journal, Action Spectrum for Melatonin Regulation in Humans, Evidence for a Novel Circadian Photoreceptor. Again, we thought this might be a career ender. I want you to take a look down in the acknowledgments. First of all, on the top line, not highlighted, you see Lane's name, Lane Brainard, because she reviews all of my manuscripts and makes them better. But what's highlighted is the inspiration for this work came from the 281 series of the Edgar Cayce readings. That's a really strange thing to put into a scientific paper when you're trying to take yourself seriously, but it was true. And why do I bring that up? Edgar Cayce said this before I was born. Right, so I couldn't have known about it in advance. He said, 500 years before the birth of Christ, Pythagoras, the first philosopher, used color for healing. Today, medical science is just beginning to see the possibilities in this method. If colors are vibration of spiritual forces, they should be able to help in healing our deepest and most subtle maladies. And in Virginia Beach at the Association for Research and Enlightenment, they have the complete works of Edgar Cayce's information that he transmitted during his lifetime. I went down there in my 20s. I spent a week. I photocopied, because that was in the era of photocopying, everything he said about the pineal gland. It filled three-ring binders, a trove of information. But he was so far ahead of the scientific community, he predicted many of the discoveries that weren't made until the 60s. So it's really a remarkable thing. Anyway, I don't want to give you the impression we were the only laboratory doing action spectra for circadian, neuroendocrine, neurobehavioral responses. And in fact, there are now 13 published action spectrum. They're shown here. At the very top, preceding us in 1996, Yoshimura and colleagues looked at mutant mice in circadian phase shifting. They found a peak wavelength of 480 nanometers, not so very far from the one we found 5 years later in humans. And our colleagues in England found also in humans in the same year that we published. Their peak was at 459. And as you go down, we don't need to beat up on the details, but a variety of vertebrate mammalian species, humans, mice, rats, monkeys, and looking at different endpoints. And to just sort of summarize this, those peaks are not identical, but they're all within 10 to 25 nanometers of each other. They're very close, and so close that there's a confidence in the scientific community that they can render this to a kind of a consensus position, and it's rendered here. First of all, let's look at the peak that's in the green-yellow range. That's photopic vision. So if you see colors, even if you have some forms of color blindness, you still have a color experience of some sort. So if you see the colors on the back end of the slide, that's your three cones in your retina working together to give you that experience of color. And that function, by consensus, peaks at 555 nanometers. In contrast, if you're looking at circadian, neuroendocrine, melatonin regulation, neurobehavioral regulation, the peak is shifted into the short wavelength end of the spectrum. Its peak, by consensus, is 480 nanometers. And there's a substantive literature to back that up. So there's a shift in wavelength sensitivity. There's also a shift in total sensitivity. It takes much less light to drive the visual system than it does to drive human physiology. Now, you notice that peak in the blue range of the spectrum is labeled melanopic photoreception. And that's the first time I've used the word, but I'm going to shift over and explain what that means. So, as I said, we thought, putting this out in the literature, we were going to be looking for new jobs. But within 1 year, it was discovered that there was an undiscovered photoreceptor in the mammalian eye, first shown in rats. And back-to-back papers by Burson and Hatter showed that. And the papers you're seeing listed there are just sort of the big-name papers. There are hundreds of papers out on this. So what you see illustrated up at the top left is the chemical structure of a molecule called melanopsin. And this particular chemical structure is human melanopsin. Melanopsin was first discovered by Ignacio Provenzio and Mark Rolog in frog skin. And they announced that to the world, and nobody responded. I mean, a lot of people worry about frog skin, sure, but a new chemical in frog skin, who cares? When Gooley showed that it existed in the human retina, then people sort of perked up and said, well, what's this? And then the next year, Burson and Hatter's group showed that it's localized, not in the photoreceptive layer of the retina, but in the ganglion cell layer. Now, at the top of the right-hand slide, you see a little diagram of the retina. And at the top are rods and cones labeled R and C. That's where most of the work of the visual system is done. And then there are a few cell layers, and then the ganglion cell layers send information off to the brain. 97% of the ganglion cells are doing the work of the visual system, allowing us to see the world. 3% contain melanopsin and are capable of detecting light completely independently of the rods and cones. Not that they don't talk to each other under normal circumstances, but the major signal is being generated by that molecule picking up photons predominantly in the short wavelength end of the spectrum to drive circadian rhythms, hormone production, and a whole bunch of other things. So melanopsin is where the term melanopic comes from. And if I was going to summarize our physiology in this moment in time here in 2024, two major pathways coming from the eye: primary optic tract for vision, retinal hypothalamic tract for biological and behavioral regulation. There are a whole bunch of acute effects of light on biology and behavior. I've talked a lot about melatonin secretion, but included are body temperature, secretion from the adrenal gland, control of the heart, alertness, your ability to stay awake through this mind-numbing presentation, brain blood flow, EEG responses, and so forth. Many things happen fairly quickly. And then there are longer-term effects: circadian phase shifting and entrainment, regulation of sleep physiology, and the putative benefits of light therapy. So that's where we are. I don't think there's anything controversial anybody would argue over in the scientific community on that, and that's a picture in time. There are applications to this. It's interesting physiology. I know everybody stays up worrying about phototransduction from the retina and what's that all about. But how do you use it? And it's been applied. The major use, the most studied use, is treatment of winter depression, seasonal affective disorder. It's also been used successfully to treat non-seasonal depression, certain sleep disorders. Newer work is showing effects on menstrual cycle disturbances, eating disorders, and comorbidities of senile dementia. So those are a field of clinical applications that derive from this very fundamental basic science. And then there are non-clinical things, domestic applications, resetting the clock after jet lag or after time change, shift work adaptation, alerting and activating effects, and sleep onset and consolidation. So there are pragmatic applications to this basic science. But it tells us that we have an emergent field of medicine that doesn't have to rely solely on pharmaceuticals. Not that I don't like to have my pharmaceuticals. Nothing like penicillin when you need it. But light is being used as a therapeutic intervention on an increasing level. So now I'm going to, applications. NASA helped support that basic work because they had a goal. They knew they had a problem. They had multiple problems with human spaceflight. But before I go to those problems, I'm just wondering, does anybody recognize this astronaut? I mean, a little hard to see behind the mask, but it's an iconic picture. To me, this picture emblemizes the heroism of the astronaut corps. This is the first U.S. astronaut to leave the spaceship and do what's called an EVA, an extravehicular activity, or a spacewalk. This was Edward White in 1965. He went outside the spaceship for 23 minutes. I'm not that guy. But I'm glad that there are people that are willing to commit to this adventure of space exploration, and we benefit in many, many ways here on Earth from that exploration activity. But as I mentioned, NASA was interested in us fooling around with the action spectrum because they saw an opportunity. Among the many things that can go wrong during spaceflight, there are disruptions of the circadian system and sleep. And NASA tries to schedule for 8 hours of sleep during the day, and that's pretty normal for this grade A healthy crew of men and women. Here on Earth, that's what they get. Studies have shown that astronaut sleep aboard the space shuttle is a little under 6 hours and a little over 6 hours on the space station. Chronic partial sleep loss, and those small numbers, I'm sure there are a bunch of people in the room that average 6 hours of sleep. So what's the big deal? If you're an astronaut and you normally get 7.5 to 8 hours a day, if you're now compressed down to 6, that's chronic partial sleep loss, which we can live with that. You can do an all-nighter. You can cut your sleep for a little while. But day after day, week after week, month after month on board the space shuttle, or space station rather, some of those flights are as long as 6 months to a year long, and the trip to Mars will be even longer. So chronic partial sleep loss is really a significant concern. Currently, NASA has used the military approach. So you give the astronauts sleeping compounds and alerting compounds. You know, sleep on demand, alert on demand. What could go wrong? So even with data showing that 71% of the crews on Space Shuttle took sleeping medications and 75% on space station, they are still getting sleep loss. So pharmaceuticals can't really reverse this problem, and they were hoping for a built-in lighting countermeasure. So let's jump out to the space station. Oh, it's just a Saturday afternoon here in Philadelphia. Here you're in the Destiny module in 2001. Coming through the hatchway is the astronaut Scott Horowitz. He was the pilot for the space shuttle that was docked at that time. And in the foreground is a flight engineer, James Voss. But what I really want to show you, and I really wish my pointer worked, but I'm going to see if it does, because maybe it's had a bit of a rest. Up here, you'll see a fluorescent light here, fluorescent light there, a fluorescent light there, and a fluorescent light there. That's the original lighting system of the space station. And as you came down this module, you'd have a set of 4 more. So that's what's providing lighting for the space station for the majority of its lifespan. There was the opportunity to create a spectrally tunable dynamic solid-state lighting system to replace that original system, and it's shown here. The prototype was developed at Kennedy Space Center in combination with Bionetics and Lighting Sciences Group. The engineers that developed it knew about the physiology we've been talking about this afternoon, that there are 2 main pathways coming from the eye that regulate biology and behavior and vision. At Johnson Space Center, here at Jefferson, at Harvard, and MIT, studies were done to see if this potentially could work. And so while those ground studies were being done, other things were being done to create the final prototypes. And here you see a photograph of the 3 different major settings. The top one is general vision, and like its name implies, it's for the astronauts to navigate the environment and see the work they're doing. Then there's one that's brighter and shifted towards the blue part of the spectrum, containing more of those wavelengths that would stimulate melanopsin. And that's intended as an acute alerting stimulus or a circadian phase shifting stimulus. And finally, there's a pre-sleep setting, which is reversed. It's much dimmer, and it's shifted to the long wavelength, red appearing end of the spectrum. All of them appear white to the eye, but they're engineered so that the underlying pattern of wavelengths is adjusted accordingly. I want to show you one other technology, and it's the crew sleeping quarters. And on the left, you're seeing a crew sleeping quarters, and it has a red tag hanging off of it, and that's because it's been certified for flight, and it's about to be flown up to the space station for installation. In the other picture on the right, you see Peggy Whitson, one of my favorite astronauts, with her foot inside the crew sleeping quarters, one outside in node 2. And there are 4 of them. And you get to have a crew sleeping quarters by seniority. If there are 6 crew members out there and you're number 5 or 6, you just have to go Velcro yourself someplace. That's how they sleep. But the astronauts really like the crew sleeping quarters. They're sound attenuated, and it's like an individual little cubicle that they get to have for their entire mission. I'm going to show you this. This is Sunny Williams. I'm going to hope that the video works. She will demonstrate the crew sleeping quarters for you.
Hello, I'm Sunny Williams. I'm up here on the International Space Station. So this is Node 2. This is a really cool module. Of course, most of these modules you'll see they have 4 sides, and they're put together. That way we could sort of work on a flat plane, either a wall, a floor, another wall, or the ceiling. But, you know, again, all you have to do is turn yourself and your reference changes. The reason I'm bringing that up is because this is where 4 out of 6 of us sleep. And so people always ask about sleeping in space. Do you lie down? Are you in a bed? Not really, because it doesn't matter. You don't really have the sensation of lying down. You just sit in your sleeping bag. So here's one sleep station right here. I'm going in right now. You can follow me if you want. So I'm inside. It's sort of like a little phone booth, but it's pretty comfy. I've got a sleeping bag right here that we sleep in, so we don't have sort of like a little bit of a cover. We don't fly all over the place. But you know, you can sleep in any orientation. I have it sleeping, feeling like I'm standing up right now, but like you saw, I'm on the floor. But it doesn't matter if I turn over and I sleep upside down. I don't have any sensation in my head that tells me that I'm upside down, so it really doesn't matter. The sleep station is also like a little office. We've got a computer in here. As you can see, we've got a couple little toys. I've got some books. I've got some clothes and other things that make it sort of like home. I'm coming out. And just for reference, that's one sleep station. This one's another, right here. There's one on the ceiling, if you want to call it, right here. And then there's a fourth on the other wall over here. So all of us sleep in a little bit of a circle. All right, come on back. There's more to show you.
So that's the crew sleeping quarters, and this is a photograph. On the left is a good friend and colleague, Bill Coyle. He's not a scientist, he's a master carpenter. We flew him down to Johnson Space Center, and he's collected here with Charles Bowen, Kevin Dries, and Jim Maida, and they're standing in front of a real crew quarters. Bill measured that up to precision and came back to this region and built us a precise replica of this, so we have this in our laboratory for doing studies, and seated in there right now is Kathy Major from NSBRI. But this gives us the capacity to simulate the environment of spaceflight. Unfortunately, Jefferson's microgravity equipment has been on the blink, and so we do this in Earth strength gravity. But if we had microgravity machines, I could stop writing grants and just sell rides in there. But that's not going to happen. Anyway, we can test the visual system of astronaut-aged individuals. We can test melatonin suppression. We can test many other biological and behavioral effects in a precise replica of the space station, at least a component of the space station. Now, here's another favorite astronaut, Kate Rubins. Dr. Rubins is a biochemist from the University of California, and her true claim to fame is that she's the first human being to sequence DNA during spaceflight. Pretty big deal. Most of the astronauts have these advanced types of skills and are able to do unique things. I like Kate because she learned that our first batch of lights were being sent out on SpaceX. 10 lights were being sent out on SpaceX for us to do a study on astronauts, and it wasn't going to be Kate. I couldn't show you her picture if it had been Kate. But she had heard about these lights. She wanted to experience them. So she made the request if she could unpack one and install it in her crew quarters. And like any good astronaut, she didn't wait for permission. She unpacked it and installed it in her crew quarters. Here you see her just before doing that, spinning it in microgravity, which caused mission control all kinds of heartburn. But she installed it successfully, and she was so happy with it before she left on her mission, she installed the other three in the other three crew quarters with permission. So this gives us the ability to begin studying the effect of this tunable lighting system on during spaceflight on the space station. And by now, here 2024, 99% of all of the original lighting system has been switched out. So the experiment is on. This is the general pattern. This slide I borrowed from Jonathan Dorey, Chief of Habitability and Human Factors. The astronauts have their period of darkness in their crew quarters or wherever they're housed. They get up, they go to the alerting form of the light, like a lighting form of cup of coffee. Then they start their general workday under general vision, and they end their day with a pre-sleep stimulus that is much dimmer and depleted in the blue part of the spectrum. This shows you the astronaut Tracy Caldwell in one of the astronauts' favorite parts of the space station. It's the cupola. It was designed by the Italians and installed, and it has a whole set of windows you can see out, and many astronauts will choose to spend their free time there because you get spectacular views of the Earth. In this study we are completing on 7 astronauts. We're looking at measurements of light on the station as well as circadian entrainment, melatonin, sleep variables, vision responses, alertness, and neurocognitive responses. I'm just going to show you the lighting metrics today. Here you see on the left is a handheld spectrophotometer. I wrote 3 separate grants to get a photometer onto the space station. All of them were rejected. But when they supported us to do a study on the astronauts, I had it. I said, look, you're not going to have us do a study and not measure the independent variable. We have to have a spectrophotometer out there. And they agreed, and they sent one out, and this is it. And here is a measurement. On the top left is a measurement of light coming from the window of the cupola. That looks like just daylight, rich in the blue end of the spectrum, depleted in the red. The GLA is the original fluorescent lighting system. The SSLA is the phase shift, blue enriched, brighter stimulus. General vision is shown on the bottom left. The pre-sleep and an even dimmer pre-sleep is shown in the next 2 components. So those are actual light measurements taken from the space station, not published yet. And then you can look at this, and this is going to fascinate everybody in the auditorium. I looked at this and I said, you should probably throw this slide out. And I said, no, they're going to really want to see this. Here are the metrics of a single measurement for general vision and the pre-sleep with shade. And I've highlighted the melanopsin IPRGC. That's the ganglion cell that contains the molecule melanopsin. You can see for general vision, the number is 336 lux, and for pre-sleep, it's 9.7 lux. I'm going to ask you to just try to remember them vaguely, because we'll circle back to that in a moment. But we can look at how much light is there to stimulate the rods, the cones, how much irradiance, and how much photons. But that's for another day. Okay, so why? Let's be truthful about this. And I'm going to ask you a question, so I want at least 3 answers. And if the first person gets it right, we'll stop right there. In the history of mankind, among all the spacefaring nations, Russia and China, India, the United States, Europe, how many humans have gone into outer space from the beginning of the early 60s to here in 2024? I've got a 200 up here. Much more. Could you be more precise? I've got an 800, 900 back there. One more brave soul. 1,020. Okay, you guys are pretty good. I checked the great source of all knowledge, the internet, this afternoon, because the number changes all the time. Flights are going up all the time. People who have gone into outer space is around 660 across all of history. Ones who've orbited the planet is a little less, because you can go up and come right back down, as some have done. So the point is, it's kind of a niche application, right? Probably those of us in the room, not many are going to go into outer space, so why would I worry about this? By contrast, from Center of Disease Control, in 2019, looking at the United States only, More than 35 million humans went into hospitals for extended stays. So you have a circumstance where the lighting, the environment, can disrupt sleep and circadian rhythms. It's been well proven. Some of you may have experienced it directly being in a hospital, and some of you might have been bulletproof. But there is a much bigger cohort of humans that go into hospitals that might have sleep and circadian disruption. So, can we take what we learned from the space station and apply it to hospital lighting? And we have been interested in looking at specifically circadian, sleep, and mood disruption in brain injury. Mild traumatic brain injury or concussion, and more serious brain injury, ischemic stroke and hemorrhagic stroke. And about a block and a half from here, there's an ICU unit that has been retrofitted with an even more advanced tunable lighting system than is on the space station now, because so much has been learned from what we've been able to do with the space program. And the little graph on the left shows you sleep onset latency. How fast is a person able to fall asleep? For a control subject, it's about 10 minutes. You go to bed, if you're a normal, healthy control person, and these are age and sex matched individuals, 10 minutes, maybe 15 minutes. You can see in the stroke patients, it's way above 25. So sleep onset is challenged under the circumstance of stroke. Can we improve on that with a built-in lighting system, as we are attempting to do for the astronauts in outer space? Okay, if I haven't said enough, I just wanted to show you the candle study. And then we'll wrap it up and we'll do question and answers if there's time. So, candlelight. From high-tech, outer space tunable LED lighting to garden variety candles. Having aged a bit myself, I sometimes wake up in the middle of the night and I can't go back to sleep. That's just a hazard of getting slightly older. For me, anyway, not everybody. So what I do is I go downstairs and I light candles in the dining room. This shows you my dining room table. Those candles were carefully prepared by Lane and the audience. And I have a candle journal, and I keep track of what happens during these experiences, including designing experiments about candles and thinking great other thoughts about candles. So, oh yeah, I've got, like, this is my fourth one, journal that I filled. So, the Nova Institute in Baltimore funded this study. That's a philanthropic funding. And we know that there's melanopic photoreception and photopic photoreception. We wanted to test the efficacy of candlelight for melatonin suppression and supporting vision. So here's our protocol. 3 nights on the experiment. 1 night, the volunteers are in darkness only. They come in at midnight, and they're in full darkness. A blood sample taken at 2 o'clock, and then they stay in darkness, and a blood sample is taken at 3:30. On the other nights, they're exposed to 90 minutes of some sort of light. The 4,000 Kelvin LED exposure is very bright light, 10 to the 18th photons per centimeter squared per second. Candlelight is much dimmer, of course, and the candlelight exposures ranged in the 10 to the 16th photons per centimeter squared per second. We used as standard a candle as Ben Warfield could find, and it's a beeswax candle. And here you see the same light meter that was used for the astronauts is being held near the candle. And if you look at the screen of that monitor, you can see a candlelight is rich in the long wavelength end, red appearing end, and very depleted in the blue end. That's just the nature, the physical nature of candlelight. The LED exposure was to a large screen, and the candle exposures varied from 1 to 9 candles in a cubicle like this. And here are the candle data. Normal, healthy men and women. Before exposure on the control night, melatonin was high. When they stayed in darkness, melatonin remained high. When exposed to bright LED light, melatonin was significantly suppressed. The light from 1 3, or 9 candles was not sufficient to significantly suppress melatonin. So I can know, oh, and this is same data just expressed in a different way, but I don't need to beat up on it. I can know safely that when I get up in the middle of the night, I can use up to 9 candles if I want. I never do. I rarely get past 4 or 5, but that's sufficient light to work on my journal and not suppress melatonin, presumably. However, there are 2 sensory systems in the eye, and I obsess about the biological behavioral, but what about vision? So we did a thing called the Farnsworth-Munsell 100U test, same test we use for the astronauts, by the way. And can you discriminate color well by candlelight? Because this is dimmer light and shifted to the red end of the spectrum. And the fact of the matter is, bright white LED light supports color vision beautifully. 1, 3, or 9 candles does not. That FM 100 score over 200 is the clinical equivalent of color blindness. You don't see color well under this dimmer light. So it's a trade-off. If you want to have good color discrimination, you can't have it without probably suppressing melatonin. So getting the balance is a goal. One last video from the space station.
Fire, it is often said, is mankind's oldest chemistry experiment. For thousands of years, people have been mixing the oxygen-rich air of Earth with an almost endless variety of fuels to produce hot, luminous flame. You would think there's not much more to learn. Dr. Foreman A. Williams, a professor of physics at the University of California, San Diego, would disagree. When it comes to fire, he says, we're just getting started. Flames are hard to understand because they're complicated. In an ordinary candle flame, thousands of chemical reactions take place. Hydrocarbon molecules from the wick are vaporized and cracked apart by heat. They combine with oxygen to produce light, heat, carbon dioxide, and water. Some of the hydrocarbon fragments form ring-shaped molecules called polycyclic aromatic hydrocarbons, and eventually, soot. Soot particles can themselves burn or simply drift away as smoke. The familiar teardrop shape of the flame is an effect caused by gravity. Hot air rises and draws fresh, cool air behind it. This is called buoyancy and is what makes the flame shoot up and flicker. But what happens when you light a candle, say, on the International Space Station? In microgravity, flames burn differently. They form little spheres, says Williams. Space station flame balls turn out to be wonderful mini-labs for combustion research. Unlike flames on Earth, which expand greedily when they need more fuel, flame balls let the oxygen come to them. Oxygen and fuel combine in a narrow zone at the surface of the sphere, not hither and yon throughout the flame. It's a much simpler system.
The brilliant work of Ben Warfield in our laboratory on putting that together. So, I see Dr. Monty in the back of the hallway here, and I still got 40 more slides. I'm just kidding. No, I'm just kidding. Dan, I'm not going to encroach on your time. By way of conclusion, this is the CIE, International Commission on Illumination, position statement on non-visual effects of light. So this is the world's source of knowledge on lighting. A high melanopic EDI during the day is usually supportive for alertness, the circadian rhythm, and a good night's sleep. A low melanopic EDI in the evening and at night facilitates sleep initiation and consolidation. Third bullet, very important, the CIE recognizes that spending adequate time outdoors during the day is associated with better health and well-being, and that exposure to natural light is a significant causal component in these effects. Slippery slope here. What do they mean by a high melanopic? Give me a number. How high is high? Or a low melanopic. How low is low? A consensus group was convened, and they published their findings in PLOS Biology in 2022. Daytime light should be a minimum of melanopic 250 lux. Evening light should be a maximum of 10 melanopic EDI, and nighttime light for the sleep environment is 1 lux, preferably less. CIE took this consensus statement, they balloted it internationally on 44 member nations, and they have published it in a reportership in 2023. So quantitative information's coming to the people who design our buildings, our homes, our institutions, and that's going to be, I think, for the better health of humans. But here we are at Jefferson in Center City, Philadelphia, in Jefferson Alumni Hall, which is just right over on that left-hand portion of the picture. And if you go outside, you can see the daylight spectrum. It's rich in blue and less in red, and that incandescent spectrum is just the reverse. Very rich in red and depleted in blue, and it's addressing the two photoreceptor systems differentially. So many people helped on the studies I shared with you today, including with us today is Jeff Greason and others. I'm not going to go through everybody's name. We've been fortunate to have the grant support, but I would be completely remiss if I didn't give a shout out to my honey bun, Lane. Here we're sitting on the steps of the little chapel in Washington, D.C., where we got married a few days later. Lane has been a superb supporter of all of this crazy stuff we do in the lab and everything else in my life and our family. So there's that. And going back to those rich books of information, the very newest testament, God said, and God gave us Maxwell's equations, which define how all electromagnetic energy, including visible light, behaves in the physical environment. And just as it was in the beginning, there was light, and the light was good. So I think the door is open now for questions. Anything that I've left unsaid, I'm happy to try and say now, and I don't want to encroach on Dr. Monte's time, so we have about 10 minutes. Yes, ma'am.
You said something earlier about the rods and the cones that I can't remember all the details now. I know you said it a minute ago, but if someone had blindness and they didn't have functioning rods and cones, they still have some type of internal way of detecting light. I didn't really quite catch all.
Right. Thank you. It's a great question. And just to do the basics is the cones allow us to see daytime vision, so we get color, detail, and motion. The rods give us nighttime vision, so even when it's dark out at night, only moonlight or stars, you can still find your way, but you're not going to see colors. You see grays, blacks, and whites. And the melanopic ganglion cells aren't participating directly or deeply in vision, but rather sending information about biological behavioral regulation. And what happens in blindness? It goes to the question of what sort of blindness it is. If you've had a blow to the back of the head and you have what's called cortical blindness, you've disrupted the occipital lobe, you may not be able to experience a single thing, but your eyes are completely intact, rods, cones, and ganglion cells. So, but the signal, the part that reads it out can't do that. Or if you've been in a terrible accident, like a fireworks accident, lost your eyes, both globes, you now have no photoreceptors. So you can't see and you can't detect light for circadian regulation. Most forms of blindness aren't those two extremes. They're somewhere in between. And that's where it becomes a much more complex story. But I think you intuited the right answer. If your ganglion layer is intact, even if you've been stripped of the rods and cones, which can happen genetically or for other reasons, you can still regulate sleep to the 24-hour cycle. Thank you for that question.
Lighting 4 candles when you can't sleep at night. Most of us turn on the television or read a book under incandescent light. Why is it possible to fall asleep watching TV with that awful light source on when it seems like it would have the opposite effect?
So sleep is regulated predominantly by 2 things, right? One, the circadian system, which is very responsive to light and dark cycles. The other is a homeostatic system, independent of the circadian system. And as you build up sleep pressure, the longer you go without sleep, the easier it is to fall asleep, right? So eventually, if you're lying on the couch watching bright screen TV, it is going to be an alerting stimulus, and it will change your sleep physiology, but ultimately, homeostatic pressure will take over, and you'll nod off. And there are many people who prefer to sleep with the TV on. TVs generally are not as bad as a personal device that's held inches away from the face, and there's a literature that shows that, because those screens on cell phones, on tablets, what have you, are very rich in the blue part of the spectrum. It's just how the technology is made. It's a great question.
Thank you.
I saw one over here.
But I did have another question. So when you showed the candle in space without gravity, the candle flame burned blue. Is that a change in the physics of the burning?
Yes, the color of the candle flame is indicative of what you experience as the color of the candle flame is indicative of an underlying shift in the wavelengths being emitted.
And that's because of the absence of gravity and that...
Yeah, I think it's the absence of gravity leads to a different dynamic of photochemistry. Yeah, but I mean, basically, the big difference is the lack of gravity, and that then, produces a different shape, a different color combination, and so forth.
Very interesting. Thank you.
Yes, ma'am.
I have a question about daylight savings time. When the time goes back to the normal time, which is what we're in now, and we get the light earlier in the day, and it gets darker earlier in the day as the season changes, does that, I mean, it does affect your, like, that's why there's seasonal affective disorder. Is there a way to, like, be out, like, if you're outside, does that help? Or, because I know that there's these light boxes. What kind of light is coming from these light boxes that helps with seasonal affective disorder?
Those are great questions, and it's topical because it just happened, right? Generally, most people experience what just happened, going back to Eastern Standard Time where we live, is, first of all, you get that extra hour of sleep, and who doesn't like that? And then you have more morning light, and that's a positive stimulus to the circadian system. And then you have less light in the evening in the pre-sleep period, so that's going to benefit sleep latency. So most people respond positively to the change in time in the fall, and just the reverse in the spring. In the spring, there's an increased risk to cardiovascular disruption, particularly strokes. There are more car crashes. That's been statistically shown because people are fighting the clock in the wrong direction. Most people don't have a severe response to the change because it's only a 1-hour change. It's like taking a plane trip to one time zone away. But if that were 3 time zones, 4 time zones, 8 time zones away, the clock gets more challenged in that phenomenon known as jet lag. So daylight savings time falls into a category that Dr. Ronenberg calls social jet lag. And social jet lag comes to us courtesy of government regulations that say we're going to do it this way. That's being fought, by the way, in Congress. There are 4 states that currently have abolished it, and there are referendum to abolish it nationwide, but it's not come yet. So hope that helps. Oh, and light boxes. For somebody with a classic diagnosis of winter depression or seasonal affective disorder, light therapy has been shown to be really the frontline treatment for that, although those patients will respond to antidepressant pharmaceuticals. The standard recommendation is 10,000 lux of white light. It can be white fluorescent or white LED, but 10,000 lux for a half an hour in the morning. Again, that notion of morning light is a more potent and appropriate stimulus to overcome the tendency towards depression. Yes, ma'am. And this may need to be the last one. All right, my timekeepers will yeah.
How does
Okay.
how does supplementing with melatonin affect the pineal gland? Is that advisable?
I missed the start of
Supplementing with melatonin?
Oh, melatonin supplementations, right. So it's so interesting because in the United States, melatonin is available without prescription. It's available in every supermarket and pharmacy that you go to. There are generally many formulations that are made of it. Not that I'm a typical human being, but I use melatonin on a daily basis at bedtime, 10 milligrams time release. That's my preferred. For some people, that would be too strong a dose. For other people, that would be too little of a dose. I have colleagues who take 40 milligrams every night. But I do recommend, and I'm not really in the position of recommending, although since it's street legal, in Europe I'd be breaking the law by making these recommendations because there you have to have a prescription. And I'm not a qualified physician to be guiding prescription medications. But here, but I think time release is very important, and it's on the label, and it's valuable because if you take non-time release, what you're doing is you're spiking the body with an enormous load of melatonin that's metabolized within about 30 minutes, whereas time release sustains it like the gland does over the nighttime. Okay, we have such a treat. Dr. Monty is coming to tell us about his stuff.